Hello. Hello, my name is Rujan Patel, and this is my presentation on my critique of my paper, The Nascent RNA Binding Complex, SFiNX, licenses piRNA-guided heterochromatin formation. So the research question and hypothesis that this paper presents is fairly simple. piRNA transposon silencing is known to be PIWI mediated. And there are a few proteins that are identified as essential cofactors in this pathway, but only one, piRNA-guided, is identified as actually being part of the silencing. How it does so is currently unknown, so therefore the question is, how is piRNA-guided connected to binding to the nascent RNA or to chromatin effectors and the chromatin itself? So the nascent RNA being the RNA that is being transcribed from the transposons. So from this question, we get the hypothesis that piRNA-guided is part of an interdependent protein complex, where another component, later identified as NXF2 NFT1 complex, is responsible for interacting with the nascent RNA, while the piRNA-guided itself recruits the chromatin effectors. So what interacts with piRNA-guided? Well, if we do a co-immunoprecipitation assay, we see that NFX2 and NXT1 both strongly interact with piRNA-guided. NXF2 is part of the NXF family of mRNA export receptor proteins, so these proteins export mRNA out of the nucleus. NXT1 interacts a lot with NXF1, which is the primary mRNA export protein. So it's not surprising that NXT1 associates with a paralog of NXF1, in this case NXF2. And PIWI interacts with piRNA-guided, but only weakly, as you can see that unbound and immunoprecipitated fraction of PIWI seem to be the same. So now let's look at what the role as NXF2 might be. So one experiment that they did is they created loss of NXF2. That did not affect the piRNA levels whatsoever, but there was a loss of transposon silencing. So what this means is that NXF2 likely does not affect PIWI binding to piRNA, but it does affect how PIWI induces the transposon silencing. So now to understand what level of control NXF2 has, they used lambda N fusion proteins. So what happens is they add this lambda N, they create these lambda N fusion proteins that allow the proteins that otherwise wouldn't be able to bind to RNA or DNA directly to bind to certain nascent RNAs or DNA sequences directly. So both lambda N piRNA-guided and lambda NXF2 were created, and they cause silencing even though PIWI was knocked out. There were other proteins that are known to be part of the pathway that they did not silence when bound to the lambda N domains that bound to nascent RNAs. So what that means, as you can see in the graph to the side, that no matter if the piRNA-guided or the NXF2 were tethered to either RNA or DNA, they showed loss of GFP intensity, meaning loss of transposon transcription. Whereas for the other proteins, for RNA, there was much lower, most of them none, HP1A much lower, still there, but compared to the piRNA-guided and NXF2, basically insignificant. But when there was DNA tethering, those three proteins did just as well as piRNA-guided and NXF2. So how do piRNA-guided and NXF2 work together? So if you mutate either of them to prevent them from interacting, the level of the other protein is reduced, even though the level of mRNA expression is the same. And so what that implies is that the transcription and translation is not dependent on the presence of the other protein, but rather the stability of each protein is dependent on the presence of the other, therefore interdependence. So, doing a size occlusion chromatography, they were able to isolate a stable protein complex that was formed from panoramics, NXF2, and NXT1. So now to really understand how both parts work in the whole, they created variants of panoramics and NXF2 that did not interact with each other and were independently stable. So again, they did the lambda N variants, and they found that NXF2 could not silence transposons. The variant NXF2 could not silence transposons when tethered directly to the RNA or DNA, but the variant lambda panoramics could when tethered to DNA. What this implies is that the panoramics is what does the actual silencing, whereas the NXF2 is what actually binds to the RNA. So NXF2 functionality, it is analogous to NXF1. Well, it has analogies to NXF1. So one of the analogies is a nucleoporin binding site, but it's concealed to prevent its functionality. So this shows that NXF2 is evolved from the NXF family. It also does have an RM and LRR fold similar to NXF1. That's where the mRNA binds in NXF1. So it implies that this is where the piRNA would bind in NXF2. There are many interactions between RM and LRR in NXF2 to panoramics. So why is this important? Because NXF1 requires lots of proteins to be in a complex with it in order to prevent just any mRNA from binding and only allow mRNAs that need to be transported out of the nucleus to bind to the protein. So in the case of NXF2, without any sort of regulation, any piRNA would be able to bind, actually any RNA, not just piRNAs. So it is likely that panoramics helps regulate this. There may be other proteins, some of which are part of the mRNA export pathway that do help as well, but that's more so a hypothesis and nothing that has been tested so far. One thing to note is that the panoramics domain that binds to LRR is part of the degron. This is the area that starts degrading when it doesn't bind to NXF2. So this not only helps with regulating what binds to NXF2, but it also helps in keeping the panoramics stable. So the significance of this paper is now we better understand how PE-mediated transposon silencing occurs. We know that panoramics actually is the one that recruits the chromatin modelers to actually silence the transposons, but the NXF2 NXT1 heterodimer stabilizes it, and the NXF2 is what binds it to that nascent RNA in order to find the transposon to silence. By tying this pathway to an mRNA export pathway as well, we now have another avenue to investigate this silencing mechanism. So to evaluate the paper, some of the good parts, this paper is well laid out, easy to follow, and explicitly talks about experiments that did not turn up any leads. Why is this important? Because even nulls are important. In fact, one of the big experiments that did not turn up any leads is that PeWe does not seem to directly interact with NXF2, at least not when it was just bound to piRNA. So I'll get back to that a little bit when we talk about future experiments. The paper also offers additional hypotheses and suggests concrete next steps for evaluation, such as next experiments. Another thing is this entire paper was looking at a Drosophila model, but throughout the entire thing, it compares the model to humans, allowing an easy way to potentially do an experiment to see if there's an analogy between the Drosophila model and humans. So an issue that I found is there's a lack of statistics, and what is often shown is not significant. As you can see, as you might have saw in the earlier slide, some of the confidence intervals crossed the 1 line, which would indicate insignificant results. This means that they either need to do more replicates or they need to use a different statistical model, or these results really are not significant. So what are the future experiments that can be done? Well, as I said, there's no direct link between piwi and the SFiNX complex, but based on the model, we see that NXF2 likely interacts with piRNA. So this has to either occur when piRNA is bound to the target RNA, when the whole piwi piRNA complex is bound to the target RNA. What the paper suggested is that this binding causes a different RNA structures, a loop or something, that and this structure is actually what allows the SFiNX complex to bind. So I would hypothesize that the SFiNX complex interacts specifically when there's a piwi-piRNA target RNA complex. You needing the piwi and associated proteins to mediate the transfer of the piRNA from piwi to NXF2, and also opening up that fold to allow that piRNA to enter. So how would this experiment work? Well, it would be 3 RNA IP on chips. So these chips would have RNA probes resembling the nascent RNAs of piwi-suppressed transposons. So the first one would be exposed to nuclear contents of ovarian cells that have NXF2 and piwi tagged. The reason for ovarian cells, as we know, those are cells that will be undergoing reprogramming of these transposons. The second RIP chip would be the same, but this time the cells are piwi negative, so it's knocked out. Third one would be a control where piRNA expression is repressed. This is just to see if NXF2 interacts with the nascent RNAs by itself. So what do we expect? Well, if the hypothesis is correct, the second and third chips would show no presence of NXF2, while the first chip would show presence. So the control being negative implies that NXF2 can't interact with target RNA without piRNA. The second chip being negative implies that NXF2 can't interact with the target RNA without piwi. The first chip being positive implies that it can interact with the target RNA in presence of piRNA and piwi. The first chip by itself does not prove that both are needed, but if you add the control and second chip in, then you reach the conclusion that it is likely needed. The presence of both piRNA and piwi are needed for NXF2 to interact with the target RNA. Here's the paper, and I hope that this presentation was informative.